Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com, Akoya Biosciences, and Oracle Bio webinar titled A Ready to Analyze High-Plex Spatial Signature Development Workflow for Cancer Immunotherapy. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Aditya or Adi Pratapa, Senior Data Scientist at Akoya Biosciences, and Dr. Lorcan Sherry, who's the Chief Scientific Officer at Oracle Bio. And together, they're going to present a new workflow for analyzing multiplex immunofluorescence images. And with that, I'm very pleased to welcome the first speaker, Dr. Adi Pratapa. Adi, thanks so much for joining us today. And the floor is yours whenever you're ready.
Thanks, Liam, for the introduction, and thank you everyone for joining us. Before we talk about the analysis workflow, I'd like to first tell you why we are interested in High-Plex-based spatial signature development. Here's the T cell. T cells are like soldiers that help fight infections and other diseases, including cancer. However, cancer cells are smart. They can escape this T cell attack by expressing a protein called PD-L1. PD-L1 works like a stop sign to inactivate T cells, and it works by attaching to receptors on T cells called PD-1 and B7-1. Both PD-1 and B7-1 can inactivate T cells. And so clinical development of PD-1, PD-L1 agents is at the epicenter of immuno-oncology drug development, with over 5,000 clinical trials as of 2021. But how do we know what patients this works in versus who it doesn't? And what do we look out for when we are guiding patients towards this therapy? There are several assays to make this decision. One particularly prevalent method for gauging the activity of PD-1, PD-L1 within tumor biopsies is through the technique known as immunohistochemistry, or IHC. IHC uses an enzyme-linked secondary antibody that generates a visible color change. Here, you can see it in brown. An IHC enables us to visualize and analyze the presence and levels of PD-1 and PD-L1 proteins within the tumor tissue. If a tumor lacks PD-L1 or exhibits high levels of PD-L1, this information is crucial. In fact, the US FDA has approved it as a companion or a complementary diagnostic for anti-PD-1, PD-L1 immunotherapies. So what other options do we have? You could look at mutation burden, or mRNA expression, or a combination of both. But how good are they in predicting patient response? This is precisely what the study from 2019 wanted to look at. They studied the clinical trial data from over 50 anti-PD-1, PD-L1 clinical trial cohorts, covering over 8,000 patients from these studies, and found that all the existing techniques or their combinations are not sufficiently predictive. Moreover, all three, IHC, gene expression profiling, and tumor mutation burden through next-gen sequencing, demonstrated comparable areas under the curve in predicting response to anti-PD-1, PD-L1 treatment. But the AUC was only about 0.7 even for the combination. So, can we do better? And what other options do we have? One of them is using something like multiplexed immunofluorescence imaging. This gives the advantage of being able to see the tumor heterogeneity while also providing single cell resolution. Unlike immunohistochemistry, immunofluorescence uses a fluorophore conjugated antibody, so you can measure the protein. What's good about it is that it can be multiplexed. And as the name suggests, in multiplexed imaging, we can measure multiple proteins simultaneously. And this study was done using PerkinElmer's Vectra instrument, which is now part of a Akoya product portfolio. In the study by Bernie Fox lab at Providence Cancer Institute, in 2017, they looked at the immune infiltrate at the tumor invasive margin in oral squamous cell cancer. When they simply counted cells in each tissue category, that would be cells in tumor versus cells in stroma, the results were very loosely correlated with patient survival. Very interestingly, patients with more FOXP3 positive cells, the immune suppressive Treg cells, had better survival rates, which did not make much sense. Then they started thinking about the spatial distribution of the cells. When they looked at the cytotoxic T cells and PD-L1 together, they observed that the number of CD8 T cells that were within 30 micron radius were indicative of how patient would survive. Suddenly, the picture became much clearer, and a low number of interacting cells predicted a better outcome or a longer survival. And this makes sense because there are more active CD8 positive T cells, resulting in a better survival. So it was not only important to know what cells are present, but where these cells are present in relation to each other. And this was reiterated by the study from 2019 I mentioned earlier. They found that multiplex protein spatial phenotyping was the only biomarker to show predictive accuracy above the 0.8 AUC threshold, deemed to be a measure of excellent diagnostic accuracy. In fact, even when biomarker modality combinations were explored, their AUC did not reach this threshold, demonstrating that the spatial phenotyping provides the highest predictive value. At Akoya, we are working hard to make spatial phenotyping available to everyone through our suite of solutions spanning the spatial biology continuum. On one end, we have the PhenoCycler Fusion, which is ideal for discovery work and for running ultra-high-plex panels with over 100 biomarkers per slide. And on the other end, we have the PhenoImager Fusion and the PhenoImager HT, which is ideal for translational and clinical applications, where one can apply targeted panels for large clinical cohort samples at a higher throughput. In today's talk, we'll focus on a scalable workflow for data analysis for Imager HT, but when we are running up to 100 slides per week, the marker selection also plays a key role. For example, one would perhaps like to study the tumor microenvironment for better stratification by looking at whether the tumor is hot or cold, or where the immune cells are located, and if the lymphocytes are activated, and so on. We offer solutions to answer these questions in the form of targeted panels. The immunocontexture panel to study if the tumor is hot or cold by studying the signs of inflammation with T cell infiltration. The immune profile panel to study where the immune cells are in the tumor microenvironment. The activated TIL panel to study if the tumor infiltrating lymphocytes are activated and if the tumor cells are proliferating. The T cell exhaustion panel to study the presence of T regulatory cells that can contribute to an immunosuppressive TME. And the M1M2 polarization panel to study the polarization of the tumor-associated macrophages. And these are available via a suite of targeted panels we call the PhenoCode signature panels. And as I mentioned earlier, in total, there are 5 panels. Moreover, each panel has been designed as a 5-plex with the option of adding on a +1 as the 6th marker. This 6th marker can be any of the markers we offer or an antibody of your choice. For example, for the immunocontexture panel, where you can study if the tumor is hot or cold using a combination of markers, you could add on CD4 to ask for where the helper T cells are. With PD1 as a +1, you could ask if the T cells are exhausted, and markers to study which cell types are proliferating, where the B cells are, or any other marker of your choice. This way, the PhenoCode signature panel let you map extra phenotypes by easily swapping out a single marker. In this example, when the PhenoCode signature IC panel is combined with CD20, you can analyze the B cell density. And when combined with PD1, the ratio of PD1 positive T cells to PD1 negative T cells within the tumor microenvironment can be examined to determine the T cell exhaustion. Moreover, the results from 5-plex base panel data is not affected by swapping in a different 6th marker. This shows the reproducibility of the PhenoCode signature panel for 3 tissue microarray slides comprising of 41 FFPE lung cancer samples with 2 different markers, PD1 and CD20. The same markers in different PhenoCode signature kits were run on serial sections to demonstrate reproducibility. The data also shows that there are no significant differences from single marker analysis in the responder or non-responder cohorts. That is, when you look at the CD8, CD68, and FOXP3, They all displayed similar cell densities between the responder and non-responder cores within the IC stain panels. Moreover, the reproducibility is not impacted by swapping the open channel marker. As another form of marker specificity within the different PhenoCode signature panels, we looked at the distribution of CD4 and CD8 positive T cells. Now, CD3E antigen is a highly specific marker for T cells, and it's present on both CD4 positive T cells and CD8 positive T cells. In this experiment, we looked at the cell densities of CD8 positive and CD4 positive T cells. For the CD4 positive cells, we looked at the cell densities of CD3E positive or CD8 negative cells in both the responder and the non-responder samples, which represents the CD4 positive cell population. We also looked at CD4 positive cells using the CD4 as a +1 marker. And you can see that the cell density plots show similar densities for the CD4 positive cells and for CD8 positive cells when calculated using different phenotyping markers. This data highlights the specificity of the assay and confirms the staining accuracy of multiple colocalized markers. In addition to this, as part of the validation process, we also tested multiplexed immunofluorescence stains for equivalence to chromogenic IHC. The PhenoCode signature IC panel is displayed here with their antibody opal pairing and the order of opal disposition. The PD1 paired with opal 520 was added to the base 5-plex as a +1, and you can see that the representative images for each marker showing the comparable staining patterns to chromogenic IHC as scored by a pathologist. Also shown on the extreme right for both panels is the average pixel intensity and the average top 3200 pixels for each marker determined from 5 regions of interest for each lung cancer tissue. And all this demonstrates consistent staining across tissue sections. By providing the base panel of 5 markers that has been validated and optimized for the PhenoImager workflow, the PhenoCode signature allows for the addition of a 6th marker of your choice to rapidly develop and optimize a 6-plex panel. Based on our own internal data, the PhenoCode signature assay development and optimization is at least 3-fold faster than the process of custom building a 6-plex assay. This offers a great advantage for projects that have short delivery timelines and lowers the barriers of expertise needed to develop 6-plex assays. As I mentioned earlier, the PhenoImager Fusion and the PhenoImager HT both allow whole slide imaging with 7 colors, that is the 6 PhenoCode markers plus DAPI. They both allow bright field whole slide scanning and offer single cell resolution, and HT does this slightly faster. The main difference, however, comes from the fact that the Fusion is more for standard throughput and is PhenoCycler compatible for ultra high-plex assays, whereas HT is ideal for high throughput and offers touchless automation for up to 80 slides and offers up to 9 color multispectral imaging capabilities. For today's presentation, we will focus on the improvements and the data analysis workflow for data obtained on the Imager HT. Not just on the panel development side, but we have made several improvements on how the data is obtained using Imager HT. In our old acquisition workflow, for those of you who are familiar with it, it starts by scanning the slides and importing them into the PhenoChart and unmixing using Inform, and then using either Inform or a third-party software for analysis. But this process was slow and took anywhere between 30 minutes to over an hour per slide. With the new HT 2.0 upgrade, we brought the unmixing on board with the instrument. With this, the unmixing time is now reduced to just 8 minutes per run. And we did a recent webinar on all the details of this new workflow that is available under the Akoya Academy webinar section on our webpage. The best part about this upgrade is that the QPative file you receive from the Imager HT is now already unmixed and is analysis ready. In addition to the raw 8-bit file that you could obtain with HT 1.0, the new version also supports a 16-bit file format for higher dynamic range, and the unmixed QPative can also be obtained in a 16-bit format. While the data sizes with the 16-bit would be larger, these upgrades aim to offer flexibility to the user depending on their analysis needs. But the unmixed file still has all the advantages of the QB file format, which comes with Akoya's proprietary file compression algorithm and allows for easy storage, data transfer, and analysis. In order to help guide the analysis, we defined this arc of spatial phenotyping. Starting from the far left, the first step in spatial biology is phenotyping cells in situ. In many ways, this is a foundational element in any spatial biology study, that is to map cells within spatial context. This is the starting point, and it requires single-cell resolution. With a single-cell mapping as the baseline, we can then move into making new discoveries. It can be AI-based cell discovery to study cell morphology or spatial functional states by combining metabolic and protein expression, or cellular neighborhoods to see how cells organize themselves to drive tissue biology. And on the translational or clinical side of the spectrum, the goal is discovering spatial biomarker signatures and establishing their clinical significance. But the final analysis steps depend on the question under consideration. But this comprehensive framework captures the continuum of needs across discovery, translational, and clinical research. And now I'll turn it over to Dr. Lorcan Sherry from Oracle Bio to talk about their ready-to-analyze workflow for High-Plex data.
Okay, thank you, Adi, and welcome everyone to the second part of the presentation. So today I would like to provide my perspective on some key considerations we see at Oracle Bio in implementing a robust multiplex IF image analysis workflow. I'll also show how we are using Visiopharm image analysis software within the workflow, and I'll highlight some example analysis, data management, and spatial profiling. However, before I start, I would like to give a brief introduction to Oracle Bio for those of you who may not be aware of us. Oracle Bio is a CRO providing industry-leading image analysis services to pharma and biotech worldwide. Essentially, we receive histology images from our clients, and we use commercially available image analysis software such as Halo and Visiopharm to develop algorithms to quantify the staining and generate readouts that our clients require. At Oracle Bio, when we think of an image analysis workflow for multiplex IF studies, we think of four principal phases. These are image QC and annotations, tissue and cell segmentation, thresholding and phenotyping, and data management and spatial profiling. And in the coming slides, I'll dive a bit deeper into each of these areas. I'll also focus on how we are using Visiopharm software in steps 2 and 3, including the use of the new PhenoPlex module in Visiopharm to support thresholding and phenotyping. For this presentation, I'm going to use example images and analysis from an Akoya 6-plex high-throughput phenocode signature panel. The markers included in this panel are shown on the right. This is also the same TMA and panel that we used in our recent joint SITC poster with Akoya. Image QC is an initial fundamental step in ensuring that we end up with high-quality data. There are many factors to consider for multiplex images as shown in the figure on the left here. Also on the right are some examples of artifacts, including scan artifacts, such as a tile that has maybe been missed by the scanner, or maybe some of the tissue image is out of focus. Marker intensity, where we may have underexposure or overexposure of a marker. Autofluorescence, such as the red blood cells here, or in collagen structures, where this autofluorescence can also be picked up by lower wavelength channels. And then also tissue folds, where we have an example of a tissue fold here that is showing brightly. And if this is not excluded in the downstream analysis from the tissue, then this area here can also pick up false positive cells. QC is mainly performed via visual check of each channel layer within the image. However, this can be time-consuming and can be supported by data, for example, using histogram pixel information per channel from the tissue, such as mean intensity, the median, standard deviation, percentiles, skew, and kurtosis values. On this slide, I'm showing some example histogram data for mean intensity per marker per core. The data can be generated either using Python scripts before the image is loaded into the analysis software, or within Visiopharm, we can de-array the cores, run a tissue find app to detect each core, and then export the mean channel intensity data. We can use this data to determine an overall mean for the sample set, as shown below here, and then to determine if any individual cores are outliers from the mean, for some examples here being shown in red. We can then view these cores and decide on suitability for analysis. Working with anatomical pathologists is important within the workflow, especially when annotations for tumor microenvironment and invasive margins are required on whole section images. This ensures the data generated is coming from relevant areas present on the tissue. Pathologists can also play a key role in supporting algorithm development, which leads me on to the next part of the workflow, tissue and cell segmentation. Firstly, I'll talk about tissue segmentation. On the left here are some considerations when performing tissue segmentation, which can all have an impact on the quality and time taken to generate an optimal classifier app. Ensuring you have a set of training images that are representative of the samples the algorithm will be applied to is important. Therefore, ensure that different tissues or tumor types capturing any unique pathology heterogeneity are included in the training set. When performing your markup training annotations, ensure that you nest these as shown in the examples to the right to provide examples of ROI borders and context to the algorithm. Finally, ensure you choose an optimal neural network. In Visiopharm, we have a few networks to choose from. For tissue segmentation or more coarse features, we prefer DeepLab 3, and for more finer features or cell segmentation, we use UNet. We can also fine-tune the deep learning settings in the AI architect, including amending channel inputs, learning rate, freeze depth, and inclusion of various augmentation factors to improve the generalizability of the app performance. Once the classifier app is developed, it can be applied to cores, and I've just got 3 examples here on screen, and you can see we are getting nice segmentation of tumor and stroma across these example cores. So moving on to cell segmentation, the tumor microenvironment contains numerous cell types, each with different shape, size, and distribution profiles. And examples of the different cell types or lineages are shown here, including T cells, which are usually small and round, macrophages, which range from small to large in size. They can be circular, or they can also be more irregular, spindle-shaped. Tumor cells, especially epithelial tumor cells, are usually larger, more rounded in shape. And then we have remaining stromal cells or structures within the stroma. For all these cells, there are specific markers provided by Akoya, and if included in your panel, can help when using AI deep learning to segment the different cell shapes. Accurate segmentation will also allow for better co-localization and evaluation of biomarkers present in your panel for cell lineage. And in the next slides, I'm going to talk about how we use panel markers to segment immune cells and macrophages. So for immune cells, we usually have the pre-trained AI fluorescent nuclear segmentation app in Visiopharm. And on the left here, you can see CD8 cells in cyan and DAPI in blue. The middle image here shows the probability mask created by the AI app. And on the right, the final cell segmentation overlay showing individual cells. Now here we've just used the DAPI channel with the pre-trained app because it's efficient and quick to use. However, in some instances, you may find that you don't detect all the immune cells that you want to because they don't have a clear DAPI stain nucleus present. So therefore, you can add the CD8 channel to the neural network training channels and then include annotations of CD8 stained cells in the training markups, further train the app, and this will improve the CD8 detection performance. For macrophages, as mentioned, based on the various varying shapes, creating a nucleus from DAPI and then a simulated cytoplasm around the nucleus will not generally capture the cell shapes accurately. We therefore manually annotate macrophages across training images. We then train the neural network using 3 input channels, DAPI, CD68, and CD163 in this instance. And this creates a probability mask shown in the center. From here, then, we use a set of post-processing steps in Vis to create individual cells based on the shape and size parameters as shown here on the right. We therefore can end up with different apps tailored to create different cell lineages to be detected. The next step then is to threshold and phenotype the detected cells. Here we use a recently added module within Visiopharm called PhenoPlex. The PhenoPlex module allows the user to dynamically choose and set thresholds per marker channel, as well as to verify created phenotypes and explore the data for visual trends and insights. Now, before I jump into the module, I'd like to highlight another thing we like about working with Akoya images is the addition of an autofluorescence channel. As I mentioned previously, autofluorescence can show up in low wavelength channels such as Opal 480, which we are using for CD8. So here is a magnified region within one of the cores showing a band of red blood cell autofluorescing in the Opal 480 channel. You can see here the red blood cells have a signal intensity similar to positive CD8 staining, which can be an issue leading to detection of false positive cells. And here is the autofluorescence channel signal on its own, showing a strong band of red blood cells down the left-hand side. So within Visiopharm, we have the ability to create extra feature images. And in this example, we have subtracted the autofluorescence channel from the Opal 480 channel, leaving only a signal from the CD8 positive cells. This feature image can now be added to the main intensity output readings and used in the PhenoPlex for thresholding CD8 cells instead of using the raw Opal 480 channel. So I have a Visiopharm instance open here where I have imported cell object data from 3 cores to demonstrate how we are using the PhenoPlex module. So let me just drag that over. So here is the PhenoPlex module in Visiopharm. I currently have 1 core on screen here to demonstrate the workflow, and I'm currently zoomed into this area here on the main screen with 4 different channels shown: DAPI, CD8, PD1, and CD68. But you can open a number of images at once on screen at the same time if you wish. On the left-hand side here is the PhenoPlex workflow. So here's where firstly, we can start by choosing the different markers that we wish to threshold. I've chosen CD8 with subtracted AF value, PD1 and CD68 mean intensity data, which has been imported from the cell object data. And then by starting with CD8, we can use this interactive scroller here at the bottom to determine the lower and upper threshold. And this is a really great addition to Visiopharm. As you see, if I move the scroller, the cells that fall within the range shown here will come up with a yellow dot on the screen. So I can just take off the other markers to show the CD8. And we can see that we're getting some nice detection. I can also put on the cell overlay that came in with the object data, and this really helps to determine cell-specific staining. So once we have a threshold chosen for CD8, we can move on to PD1, and just to change the channel, I've actually pre-selected the threshold range here, so you can see the yellow dots are highlighting the cells where there's PD1 positive green present, and similarly for CD68. Again, I've pre-selected, in the interest of time, a range here that highlights those cells that have got yellow for CD68 staining. So once done, we can then check the positivity. We first save these thresholds, and we can check the positivity in the co-occurrence matrix. So here, this will allow us to see which cells are being detected by the thresholds chosen. So if I just go back to my immune cells and clicking on the button here, highlights all the CD8 positive cells. Likewise, if I click here, we will highlight the CD8 PD1 positive cells. So we can actually scroll in here and see which cells are actually dual positive staining. So in this instance, we've got 3 cells up here that have got both CD8 and PD1 present, which looks pretty good for the thresholds. We can also check to see if there are any high levels of co-occurrence we would not expect. For example, CD8 and CD68. So if I just change over to the CD68, we can see a few of those. There are a few, obviously, but when we look at them in more detail, I think these are true CD8 positive cells, and maybe the macrophages are surrounding or overlapping the CD8 cell object boundary here. So once done, we can then view the phenotypes pressing this button here, and this will then show us all the phenotypes that have been detected for the different marker thresholds that we have used. And this can be done interactively by switching on and off various buttons here to review the different phenotypes that are present. Finally, we can perform further QC and exploration by the Explore QC button at the top here. So I'll just press that. So once the data has loaded in, we have this menu of different options for data exploration, including t-SNE, scatter, box plot, and histogram. But I'll focus on the scatter plot here, which allows me to plot different data such as CD8 with the subtracted AF versus DAPI mean intensity. So when I press plot data, we get a scattergram here where each dot represents a cell in the core. And then we can also color the dots based on certain parameters. In this instance, I've chosen the CD8 intensity. And now when we click on an individual dot, it will actually highlight that cell for us in the tissue as shown. And again, this is really helpful in allowing us to understand if the threshold range that we are using is detecting cells within too high, or for example, too low a signal intensity, or that we're seeing co-localization of markers in the same cell where there should not be co-localization. So in the interest of time, I will leave it there and go back to the presentation. So once we have established our thresholds in PhenoPlex, we now apply these to each core to generate the cell phenotypes. Here we have an example from the Immune Cell app detecting different immune phenotypes in the tumor and stroma ROI. And here is some data from the example cores from the TMA that we showed in our poster at SITC. So on the y-axis here, we have the percent phenotype within the different ROIs of breast cancer and also lymphoma. And similarly, for the macrophage app, we can detect different macrophage phenotypes in the tumor and stroma ROI. And here again is some data from example cores from the TMA that we showed in our poster at SITC. So once the analysis has generated the phenotypes, we can then extend the analysis to evaluate spatial profiles. This can be done in two ways, either within the analysis software, where we evaluate parameters like infiltration analysis, cell-cell proximity, minimum maximum distance relationships. Alternatively, we can export cell object data files for post-processing outside of the image analysis software. Exported cell object data can include mean intensity, XY vector coordinates, phenotype, labels, cell size, shape, etc. And here we use programming scripts such as MATLAB and Python to interrogate data files for deeper or more complex spatial profiling. In the above example from our recent SITC poster, we exported cell object data file from Visiopharm and processed the data using a Python script to determine a range of neighborhood distance parameters as shown above. More information and interpretations can be found in the SITC poster at the Akoya or Oracle Bio website. Finally, I wanted to mention the importance of our IT infrastructure. There has been increasing demands placed on our IT group over the past few years to help support our scientists perform multiplex IF analysis. These have included providing an environment with the ability to manage and store increasing image volumes and file sizes, capable of handling complex image analysis tasks at speed and scale, to efficiently integrate new software updates and capabilities, to be easily accessible with enabled remote working across multiple locations, and it needs to be cost effective and address our green credentials. So at Oracle Bio, we have placed all our image analysis software within a scalable cloud environment, Amazon Web Services, AWS, which can be accessed by a web-based portal, putting the scientists in charge of which software they wish to use and with what CPU, GPU capabilities required for the work, their tasks that they're doing in real time. Although not directly part of the IA workflow, our IT environment is crucial in helping to deliver powerful, efficient, and scalable analysis for large multiplex IF images. So to conclude, I've given an overview of a standardized image analysis workflow that combines data-driven image quality assessment, IA scientist and pathologist inputs, the power of AI deep learning, utilization of Visiopharm PhenoPlex module, and downstream data processing. And together, this has enabled us to interrogate complex pathologies within high-plex staining panels to deliver quality data from valuable tissue samples in an R&D setting. And with that, I would like to thank everyone for your attention. We now have some time for a Q&A, so if you have any questions, please do add them to the chat box if you haven't already done so. And I'll just pass back to Liam now to get us started.
Excellent. Thanks so much, Lorcan and Addy, for great presentations here. So let's jump in with a question here for Addy. So is the unmixed QP TIFF file also normalized to account for exposure times during the acquisition?
Yeah, the short answer is yes. That information on normalization is part of the QPTIFF metadata XML file. So unlike the previous version where the unmixed file was stored as a 32-bit float, now the data is stored as a 16-bit integer, and you can read in this normalization factor from the metadata. And software like Visiopharm already support this natively, but yeah, you could always, add on that feature depending on your use case.
Excellent. Yeah, great to hear. And Lorcan, here, one for you. So where do you see AI continuing to impact the multiplex IF image analysis workflow?
Yeah. Can you hear me okay, Liam? Yeah.
Yeah.
Great,
Sounds great.
Great. So in my presentation, I demonstrated the use of AI in the sort of cell segmentation process and also the tissue segmentation process. And certainly in that area, we've seen some great improvements in how we are segmenting tissues and cells in the step before the thresholding. I think if we look at the early part of the workflow, at the image QC part, certainly at the moment, that's traditionally done by manual review, visual review, and that can be quite time consuming. It can also be subjective. And I think there's a number of companies out there, including ourselves, are looking at how we can maybe use AI to train algorithms to recognize certain features around out of focus, staining variants, tissue artifacts, and trying to automate that process so that it makes it more efficient in that part of the workflow. I think also when we look at the area of, say, thresholding or choosing a threshold for positive staining or positive cells versus negative cells for each of the markers in a panel, traditionally, again, that's done through a manual method where maybe a global threshold is used across the whole image set. And that can lead to challenges. Sometimes there are maybe some staining variances between different sections or even in different channels. When you multiply that across, say, 4, 5, 6 channels in an image, you can have you can maybe have some amendments to those types of thresholds being set. And I think here there's certainly an opportunity for AI to be incorporated in to support some adaptive thresholding, so that the AI is trained to recognize how maybe positive staining looks maybe in a low exposure background or maybe in a high exposure background. What is positive staining? Can that be detected? And also in areas where there's maybe non-specific staining or things like that. So moving away from maybe global thresholds to using adaptive thresholds supported by augmented AI is certainly something I think is going to improve the efficiency of these in the R&D setting. But certainly as we move into things like companion diagnostics and how we use this in maybe a clinical or healthcare workflow, certainly AI and supporting that adaptive nature is something I think will make that a meaningful process in that setting.
Yeah, great answer. Feels like it highlights some of the great advantages of AI, especially for image analysis. It's a really powerful thing that you can do with it. All right, so is imaging for phenocode signature assays any different in terms of the unmixing and autofluorescence removal? Eddie, I think this one's for you.
Yeah. So the guidance remains the same in terms of creating the unmixing library, both for the OPAL assays and the phenocode signature assay. But the official recommendation is that you create a phenocode signature specific library and for the tissue type that you're working with. Just to be, because that can change from tissue to tissue.
Excellent. A few questions about validation here. So, Lorcan, can you validate the algorithms that you develop? And also, what level of validations do the panels have? And you mentioned clinical trials just now. How are they used in clinical trials, if at all, yet?
Yeah, I can start there. Certainly, we can validate the algorithms. A bit more complex to validate multiplex fluorescent algorithms compared to maybe bright field chromogenic algorithms that have been developed for bright field chromogenic images. But we normally take a sort of a dual approach to the algorithm development. Firstly, we think of the tissue classifier algorithm, the one that is segmenting the tissue into different regions of interest. So if that was tumor or oncology tissue, it'd be tumor stroma regions of interest. And we will validate that using a dice coefficient approach, where we create field of view boxes on a set of validation images after the algorithm has been trained. Our pathologists will be involved in this process, where pathologists will go in and manually annotate areas that they see as tumor and stroma within these fields of view. And then the algorithm is applied to the field of views independently to create its own data. And then there's a comparison of that data using the dice coefficient to see how well the pathologist or the ground truth data aligns with the performance of the app. For cell analysis, again, as I mentioned, this is a little bit trickier. We've got not just one set of maybe readouts per marker, but we may in a multiplex environment of 5, 10, or even more. So every channel or every marker needs to be validated independently on its own accord. Here, again, we will apply field of view boxes to a set of validation images. We will go in pathologist-assisted reads of those boxes to count the numbers of positive, negative cells for each of the marker channels. And then the algorithm will be applied directly to those boxes and the data generate. And here we would perform correlation analysis to compare the readouts of the algorithm directly with the readouts of the manual review. So yeah, certainly the algorithms can be validated in that format. I think I don't know, Adi, if you want to talk a little bit about how they're being sort of validated from an assay perspective.
Yeah, To add to that, the validation data for the phenocode signature panels is provided as part of the data sheets, and you can always reach out to our technical sales teams if you need more information on how the panel validations perform on our end.
Fantastic. All right, Adi, does steric hindrance of the barcoded antibodies affect the multiplex phenocode signature assays?
Yeah, so again, this is something that we included as part of our validation during the development process, where we tested that both the single stain antibodies and also in the multiplex setting, you see the same staining and specificity. And yeah, this is part of the validation that's done to test specifically for steric hindrance.
Excellent. I think this one will be for you, Lorcan. How long did it take for you to establish your cloud infrastructure?
Yeah, I talked a little bit about this in the presentation. It was certainly something that we started about 4 or 5 years ago and has been a sort of a constant development. We pretty much started due to the need to think about image storage. Multiplex IF images are quite large image files, so trying to store those locally on a hard drive, on a sort of a infrastructure internally on site was becoming more challenging. So that was one of the reasons we moved to the cloud, to get that sort of scalable storage. But then we needed more connectivity about how our clients send us images. So again, thinking about how these images are transferred across sites and between geographical regions and so forth. We set up AWS buckets, which was another sort of benefit of moving to the cloud. Thirdly, we also needed scalable access to GPUs and CPUs. A lot of the deep learning neural networks that we use run on GPUs. There may be certain studies where we will be analyzing maybe a couple of hundred images, and some of these images may take a number of hours to analyze. So we wanted to get parallel access to numbers of GPUs through our Visiopharm software. We can analyze as many images in parallel as we wish. Access to those GPUs and CPUs in the cloud and being able to switch them on and off when needed from a cost perspective was a real benefit. And then finally, we also needed a way of our staff to be able to access the software both from a hybrid environment, from at home or from in the office based. So being able to log into a portal and access that, not necessarily be physically on site all the time, was important as well. So a lot of that was brought together in a build over time. We didn't have all the IT expertise internally in the company. We worked with one of our sister companies, Ciento Technology, who specialize in this. And certainly it's the backbone of how we operate now. For a CRO, it's about creating quality data, accurate data, but it's also about creating that efficiently. And certainly our cloud infrastructure has allowed us to do that.
Excellent. All right. This person says that they use PhenoCycler Codex antibodies. Adi, can these be used with the PhenoCode panels?
The short answer is no. Each antibody that we offer via the PhenoCode signature assay has been tested specifically for use in that assay. So you cannot use the Codex antibodies in the PhenoCode signature panels.
All right. All right. Question here is a bit long, so bear with me. But it says, great presentations, thank you. My question is, what would be the way for controlling the phenotyping correctness across consecutive sections with partially overlapping staining panels? So when cell densities don't correlate between slides, but visually phenotyping looks okay on both of them, do you need a ground truth in such a case? And if yes, in a data set of 100 slides, should it be calculated for each slides and for each marker? Do you have any recommendations for this kind of case?
Yeah, I think when we're talking about serial sections, the data from section to section will not always be identical. There's a distance between the sections, and the cell profile does change slightly. I think to gain confidence, I touched upon the validation process, and I think that's maybe where you really try and build your confidence in the performance of the apps, where you're choosing your field of views on a validation set of images, and you're doing a manual counting with pathologist interaction and assistance, and comparing that to the performance of the algorithm. And I think that's an important step. If you're really wanting to ensure confidence in how those apps are going to perform moving forward. So yeah, I would say trying to build in the validation, prior to sort of execution of your actual work samples is an important step.
Excellent. Yeah, great answer. Another one for you, Lorcan. Do you have an approach for discovering new biomarkers? And maybe you could reveal a bit on this topic, like types and examples, or any trends ongoing.
Yeah, it's interesting. As a CRO, a lot of the work that we do for our clients, especially with the Akoya panels, it will be based in the translational area, where R&D space, where certainly those clients are looking for interesting biomarker profiles in particular disease samples and so forth. So they will use the panels, and then we will analyze those that staining profile with the algorithms we develop, and we'll generate a whole set of data. And it will be dependent on the tissues that the staining and the analysis has been applied to, and the questions and the therapeutic areas that those samples are aligned with. Certainly, there's a lot of data that can be generated from samples, from individual cell population counts, immune cells, macrophages, tumor cells. You can look for phenotypes of those types of cells. So you can look at different subsets of T cells. You can look at activation status. And again, the Akoya panels are very much set up for this. We generate that data. We generate all the sort of the raw data. We can summarize that data and look for different sort of profiles. As I mentioned, there are some spatial interactions as well. That is something that you can do at a cell-to-cell level, but now it's been expanded into more neighborhood analysis and so forth, and definitely more sort of IT-intensive bioinformatic processes to look for unsupervised review of that data, as well as hypothesis-driven. So a lot of the clinical trials we will work on have their specific questions already, and we will look for data to support that. But other questions, at the translational level will be biomarker development and biomarker identification driven. And again, we can provide all the data there and send that back to our clients, as well as being able to perform the types of readouts that I talked about.
Excellent. Adi, you still there? Just want to make sure that you can still hear us. It looks like your camera dropped out for a sec.
Yeah, I'm still here. Can you hear me?
Okay, fantastic. Yeah, all good. All right, I think in the interest of time, we'll just have one last question, maybe a question looking forward to the future. Are there any upcoming developments in both your services, or maybe trends, research avenues that are emerging that you're really looking forward to? And so, Adi, maybe we'll start with you, and then we can move to Lorcan.
Yeah, I think in terms of trends, it's the data generation part of it. I think is kind of there. We can now generate hundreds and thousands of slides in a matter of weeks. And to me, how we can take advantage of all of these, just like that sort of data that's out there, in order to come up with new ways of analyzing, and also in terms of how they can be applied in a clinical setting is what excites me the most.
Yeah, great answer. And what about you, Lorcan?
Yeah, I think the area of multiplex IF has really allowed us to much better and profile the biology, the underlying biology of disease processes and therapeutic responses. If I go back 10 years ago, we were dealing with single chromogenic stains and single cell populations and trying to determine mechanistic responses and biomarkers around a sort of a single cell count readout. Now we're able to look at all the different sort of phenotypic profiles in cancers, and this is especially important in immunotherapy, which is one of the key sort of areas for cancer therapeutics at the moment. So certainly multiplex IF has allowed us to generate more data, but certainly the spatial component, and Adi touched upon this in his, as well as myself, not just the cell counts, not just how many cells are present in a tissue, but where are they present and how are they interacting with each other, is now becoming not just an interest from a research and scientific perspective, but also from a companion diagnostic and a healthcare perspective. Can we start to use data, to spatial data and contextual data of cells on tissue sections, to identify which patients will or will not respond to a certain therapy? And I think that's a really key area moving forward for us in this field. And there's a lot of validation there to be done. There's a lot of standardization in the process, from the staining, the imaging, the analysis, the use of algorithms, and the data generation. But certainly the depth of data and the different types of data that we're now able to extract from these, very precious tissue samples is allowing us to think this way. And I do see over the next, 5 years or so, great strides moving towards how we can maybe use this type of technology to the benefit of patients.
Yeah, definitely. A lot of work to be done, but certainly a lot to look forward to. But yeah, Lorcan and Adi, thanks so much for sharing your insights today. It's really been a pleasure having you with us.
Thank you. Thank you.
Thank you.
Big thanks also to the audience for participating. And in closing, we hope you enjoyed this Scientist.com, Akoya Biosciences, and Oracle Bio webinar. And we'll see you again next time. Have a great day, everyone.